I have the great pleasure now of being joined by Dr. Rob Knight, who is talking about AGA's active learning session on the gut microbiome. And you put a tremendous amount of planning into this, even asking attendees to send in fecal samples that were analyzed.
Absolutely. So a lot of members of the public are getting really interested in what can you get by profiling your gut, whether you do it with a research project like American Gut or whether you do it with a company. And we thought that it was really important to give the AGA membership an opportunity to see what those profiles look like, how they can interpret their own profiles, and also what you can and can't say about them clinically, and what you might be able to say in the future that will give you a clinically useful diagnosis out of this technology.
And that, I think, is the key, is that interpretation when you get back that information for any of your patients, trying to interpret that for them.
Absolutely, and it's such a huge problem at the moment because right now, depending on what you do, you're going to get back a list of 1,000 species or 1,000,000 genes. And at the moment, what are you going to do with that? Maybe you can refer your patient to your colleagues in psychiatry for being crazy enough to think that you could interpret it, but that's about it. And so what we're trying to do is develop new software to put that information in context so that as a practicing gastroenterologist, you can actually get something useful out of it that you can communicate to your patients. But part of the problem is not in the software, but in the data sets. So we're working with Bill Sanborn, among others, to try to figure out how can we get some clinically well-validated data sets for IBD, run them through exactly the same protocols, and then you can see your own sample or your patient's sample in the context of those clinically validated samples.
Yes, because that information, there's so much research out there to show that the gut microbiome is connected in our overall health and in so many diseases.
That's absolutely true, and 10 years ago when I started working on obesity with Jeff Gordon at WashU and then later other researchers interested in that topic as well, people thought it was crazy even to try to connect gut microbes to obesity, and then as the number of other diseases that have been linked to the microbiome has accumulated, so things like diabetes, things like fatty liver disease, even really surprising things like rheumatoid arthritis, there's a very solid story for in the gut now. And then if you look at the mouse model data, there's really solid data in things like multiple sclerosis, in autism, even in depression, to the extent that the mouse models can tell us things. And so those haven't been made really solid in patients yet, but the fact that there's a biological mechanism at all has been a total surprise, and it's just in the last 5 years that there's been an appreciation that the gut can have such far-reaching effects on the brain.
There is an explosion of information right now, but clearly people need to learn so much more. So from this right here, what you've done, what can attendees take back into their own clinical practice right now?
Well, one thing that the attendees who provided samples can take back is that they look much more like our reference population for American Gut and do not look like our IBD cases. So in that sense, that's reassuring because a lot of clinicians have been worried that maybe they're picking up microbes from their patients that could be getting into their own microbiome, and we don't see a lot of evidence for that. Clinicians can also take back the fact that in the context of a carefully controlled study, we're very good at discriminating cases from controls, and even at subtyping IBD. So, for example, colonic versus ileal Crohn's have substantially different signatures. We're making a lot of progress on the association networks of bacteria that lead to those different outcomes. But in terms of being able to look at a patient and give that patient a diagnosis, there's still a lot of work to do before we can move towards an FDA-approved test. And it's going to be important to temper your patient's enthusiasm because the public enthusiasm for this stuff has greatly outstripped scientific understanding. And explaining to them the difference between research use and what you can do in a study versus clinical use and what you can do for a diagnosis is really important.
It's such a fascinating field, but you have to temper where that information is coming from.
Absolutely. Now that and figuring out is the information you're getting scientifically valid, that's really important. And both with this and with probiotics, where there's so much public enthusiasm and so little data. So I moved here to San Diego just a little over a year and a half ago, and bought a couple of citrus trees, as one does on moving to this climate. And at the gardening store, the fertilizer for the citrus trees had emblazoned on the bag "new" with probiotics. So apparently the probiotics are, at least in concept, just getting everywhere. including to places that you might think they might not apply.
It's the catchword, but thank goodness you're doing the research to have science be involved as well. Thank you so much, Doctor.
Well, thank you so much. Really glad you're interested in this topic, and look forward to seeing you at the session.